Okay, so our next organelle we are going to be discuss is lysosome. As you guys all know, in the previous video we have discussed about the Golgi complex. So what is lysosome? Lysosome means splitting and soma means body, which means that the this is the organelle which split the organelles. Okay, so that are the lysosome. If we talk about the pH, so they are about 4 to 4.5 pH. Okay, so they are basically the single bounded organelle not, they are the vesicle. Okay, so the as you guys all know, in the Golgi complex we were like, I was saying that this is the like vesicle like structure. So these vesicle are what? These vesicle are the lysosome actually. Okay, and they contain the hydrolytic enzyme to digest the food. Hydrolytic enzyme to digest the food in the amoeba. Okay, so the lysosome are basically made up from the rough endoplasm reticulum. As you guys all know, we have discussed the rough endoplasm reticulum and endoplasm reticulum, and we have discussed the smooth endoplasm reticulum. So they are basically made from the rough endoplasm reticular, and they are basically transport to the Golgi complex, and from Golgi complex to the smooth endoplasm reticular. Their enzyme are basically, you can say, made from the rough endoplasm reticulum, then they are transported to the Golgi complex, then they are transported to the smooth endoplasm reticulum. After that, in smooth endoplasm reticular, the you can say that the what's the word I have to use, that is basically the modification. Yes, the modification of the enzyme occur, and the modification is called the vesicles. Okay, they are like the from the, you can say that the enzyme are basically, after the modification from the smooth endoplasm reticular. Okay, these enzyme are basically then will be released from the Golgi complex. The Golgi complex, we were having two parts, transphase, as well as we were having one more part, that was the cis yeah, transphase and the cis phase. So they are basically will be released by the transphase in the form of the vesicle. Okay, so the enzyme, they will be transported, they are made from the rough endoplasm reticulum, they will move to the Golgi complex, then smooth endoplasm reticulum. But in the Golgi, the modification is occur. Okay, after then they will move to the Golgi complex, and in Golgi complex the modification will occur, and they will form a vesicle. Then this vesicle is basically called the lysosome. Okay, so the lysosome, when it is not like performing any kind of a function, they are called the primary lysosome. Okay, in plants, in fungi, they are basically like in the, you can say that they're in the certain vacuoles are there. Okay, and that vacuoles are basically, vacuole is the food vacuole. For example, this is the amoeba structure, and we are having the food vacuole. What is food vacuole? In which the food is there. Okay, the lysosome will be coming, and they will be digest the food particles through the hydrolytic enzyme that I told you guys. Okay, so this is basically the carry out by the enzymatic hydrolysis. Okay, so lysozyme basically consists of 40 different enzyme, 40 different digestive enzyme. Okay, this enzyme basically having a main role to break the macromolecule into the macro means larger molecule into the smaller molecules. Okay, so the lysosome, if you talk about, so they are, their content is basically acidic in nature. They are basically acidic in nature. Okay, one more thing I want to tell you guys is that when the lysosome is start performing the function, when the lysosome is not performing the function, they are called primary lysosome. When they are starting the function, they are calling the secondary lysosome. Like they perform different pathways like endocytosis. I'll be telling you the pathways: endocytosis, phagocytosis, phagocytosis, okay, autophagocytosis. So when the lysosome is combining with the endocytosis process, we are calling that as a endosome. When the lysosome is like doing the phagocytosis process, we are calling that as a phagosome. When the lysosome is combining with this one process, autophagocytosis, they are like doing the process of autophagocytosis, so we are calling that as a autophagosome. Okay, so the primary lysosome will be forming into the secondary lysosome, in which the breakdown of the smaller molecule will occur, like amino acid, monosaccharide, nucleotide, fatty acid. We are having many kind of things. So function of lysosome is basically the intracellular digestion, that I'll be telling you what is intracellular digestion. Intracellular digestion, autophagy. What's autophagy? I will be telling you too. Autophagy, and many like you can say that sometime they also release the extracellular enzyme too. Okay, so the, as you guys all know, in the food vacuole, in the vessels, like food vacuole is there. So I told you guys there is a food particle is there inside the, like this is the food vacuole. Okay, so the lysosome will be fused with the few food vacuole, and for example this is a amoeba structure and they are in the food vacuole. Okay, with the help of the endocytosis, the phagocytosis, the food is entered into the amoeba body, and lysosome will be like fused with that, and with that the food will be become digest, by become digest, and the waste thing will be the food will be basically the food vacuole will contract and the waste particle will be moved outside, and through the, you can say that the undigested waste will be removed from the cell membrane by the exocytosis process. Exocytosis means to remove the unwanted substance. So unwanted substance will be moved toward the exocytosis process outside of the, you can say, the cell membrane. This is called the intracellular digestion, which means the digestion inside the cell with the help of the lysosome and the food vacuole. Now what is autophagy? Autophagy is basically the process in which the, you can say that the unwanted substance are basically engulfed or digest with the lysosome, for example they are like unwanted substance, they are basically, the lysosome is basically the like engulfing the unwanted substance. So that process we are calling as a autophagy, and also we are calling autophagy as a self-eating process. Okay, self-eating process, because they are becoming to digest like uh, one more thing, autophagosome. Autophagosome or autophagocytosis, this is the process in which the lysosome is like eating the his own organelle. Lysosome eating the, eating on organelles in the cytoplasm. We are calling that process as a autophagosome. Okay, so this process is called the autophagosome, in which the lysosome basically digests the cell's own organelles. Okay, sometime in the development stage, one more I want to tell you guys it, in some time in development stage, when there is like you can say that a type of death is like occur, we are calling that as the autolysis. So the cell which are like committed to be death, that is called the autolysis. And then autolysis when occurs, and then the lysosome will getting burst, and after the bursting of the lysosome, the hydrolytic enzyme, they are like present in the lysosome, so they will be, you can say that, dispersed out to the cytoplasm. Okay, in this way the like fragments will be there. So due to this function, lysosome will be getting dead, and that's why we are calling lysosome as a suicidal bags too. because lysosome will be getting burst because of the autolysis process. When the cell will committed to be dead, autolysis will occur, lysosome will be getting burst, the hydrolytic enzyme will move disperse out in the cytoplasm, so the lysosome will be getting burst. So that's why we're calling it lysosome as a suicidal bag. Okay, so lysosome are basically the vesicle which contain the hydrolytic enzyme, which like we having the function to like break down the macromolecules to the micromolecules. Okay, so this was about the some kind of a process what the lysosome basically done, okay, in the body cells. So this was about the lysosome. In the next video we will discuss about the peroxisome and the glycosome.